Now, why is that importance of the why you require that proton gradient? Okay, you require that proton gradient because you want your ATP to be produced. Okay, so you want what? 1 minute. You want what? You want your ATP to be produced. Am I clear to you? Now, where ATP and NADPH is used, obviously amongst the oxygen diffuses out of the chloroplast, and ATP and NADP helps in what, children? Leading of a particular formation of the sugars. So sugar formation ge nimge en bekagutte makla, you require ATPs and NADP. Now we'll start with your Calvin cycle. Okay, it was your Melvin Calvin who introduced the cycle called as Calvin cycle, and it is also called as a C3 cycle. Okay, where it includes particularly 3 steps. What are those? Carboxylation. Okay, first step is what, makla? Carboxylation. Second is your reduction. Third one is your regeneration. These are the 3 particular steps. Now see, children. Calvin cycle, carbon atoms from carbon dioxide are fixed and are used to form a 3-carbon sugar. So the first stable compound is a 3-carbon sugar, hence we call it as a C3 cycle. This process is dependent on ATP and NADPH. ಇದನ್ನೇ ನಾನು ಹೇಳಿದ್ದು ಅವಾಗಲೇ. Calvin cycle is an independent cycle. ಅಂದ್ರೆ ಲೈಟ್ ಮೇಲೆ ಡಿಪೆಂಡ್ ಆಗಿಲ್ಲದಂಗೆ ಇರಬಹುದು, ಆದ್ರೆ the particular products which are formed during the dependent reactions, ATP, like your ATP is formed by light reaction, is or no, light dependent. So they depend on ATP and NADPH formed from the light reactions. Okay, now so light reactions is carried out in thylakoid's membrane, while Calvin cycle is taking place where? In stroma. Taleli itkoli, light reaction ellagirutte thylakoid membrane alli, Calvin cycle ellagutte stroma dalli. Carboxylation, it includes the following stages. Okay, first one is carboxylation, fixation of carbon dioxide. Okay, where it is fixed by a particular RUBP. Am I clear to you? The rubisco. In the presence of the enzyme RUBP carboxylase, it results in the formation of the molecule called as 3-phosphoglyceraldehyde. It is the first form particular what, makla? The formed stable compound. Since it is a 3-carbon, we call this cycle as a C3 cycle also. Next one is your reduction process. Now coming to your reduction process, this stage includes a series of reactions that results in the formation of glucose. Am I clear to you? Where it utilizes 2 molecules of ATP and 2 molecules of NADPH for the fixation of how many molecules of carbons? 6 molecules of carbons. Next one, to produce your 1 molecules of glucose. Regeneration, this stage includes the regeneration of carbon dioxide acceptor molecule and requires 1 ATP. ಅರ್ಥ ಆಗಿಲ್ಲ ಅಂತ ನಂಗೆ ಗೊತ್ತು, ಇವಾಗ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. Now carbon fixation andre ಮಕ್ಕಳೇ, usually the carbon dioxide is fixed by a particular molecule. Am I clear to you? So in the presence of what? Rubisco. Okay, so which will in turn form what? The reduction. Okay, so your 3-PGA whichever is formed will undergo the reduction. Am I clear to you? So again, after your particular reduction, it will regenerate back to particular cycle. Okay, so this is how your carbon fixation takes place. 1 minute. See over here. Your carbon dioxide is fixed. Am I clear to you? Usually the mono molecule will convert into bimolecule, where you have your 3-phosphoglycerate formed. It's your first stable compound. Your 3-phosphoglycerate will get converted into 1,3-biphosphogycerate, where it will undergo deduction to form glyceraldehyde 3-phosphate. Finally, it will be for regenerating back your ribulose bisphosphate. So remember, ಮುಂಚೆ bisphosphate ಗಿಂತ ಮುಂಚೆ ribulose monophosphate ಇರುತ್ತೆ ಇಲ್ಲಿ. Okay, ribulose monophosphate will be there. Monophosphate will, where your ATP will get converted into ADP. So 1 inorganic phosphate liberate ಆಗುತ್ತೆ. So this inorganic phosphate will be accepted by ribulose monophosphate to form biphosphate. ಅರ್ಥ ಆಗ್ಲಿಲ್ವೇನ್ರೋ? ಇನ್ನೊಂದ್ಸಲ ಹೇಳ್ತೀನಿ, ಕೇಳಿಸ್ಕೊಳ್ಳಿ. ಇಲ್ಲಿ ನಿಮಗೆ ರಿಬ್ಯುಲೋಸ್ ಬೈಫಾಸ್ಫೇಟ್ ಇದೆಯಾ? ಇದಕ್ಕಿಂತ ಮುಂಚೆ ನಿಮಗೆ ಮೋನೋಫಾಸ್ಫೇಟ್ ಇರುತ್ತೆ. ಮೋನೋಫಾಸ್ಫೇಟ್ ಅಂದ್ರೆ ಏನು? ಒಂದೇ ಫಾಸ್ಫೇಟ್ ಅಂತ ಅರ್ಥ. ಬಟ್ 1 ಫಾಸ್ಫೇಟ್ ಇದ್ದಿದ್ದು 2 ಫಾಸ್ಫೇಟ್ ಹೆಂಗಾಯ್ತು? your ATP ಇದೆ ಅಲ್ವಾ, ATP will get converted into what? ADP. ATP ಅಂದ್ರೆ ಏನು? Adenosine triphosphate. Adenosine triphosphate got converted into what? adenosine diphosphate. So 3 ಇಂದ ಇಲ್ಲಿ 2 ಆಗ್ತಾ ಇದೆ, ಕರೆಕ್ಟಾ? ಸೊ ಒಂದು ಎಲ್ಲಿ ಹೋಯ್ತು? that ಒಂದು came and attached to your ribulose monophosphate. ಸೊ ಇಮಿಡಿಯೇಟ್ ಆಗಿ monophosphate ಗೆ ಅಟ್ಯಾಚ್ ಆಗಿದ ತಕ್ಷಣ ಎಷ್ಟು ಫಾಸ್ಫೇಟ್ ಆಯ್ತು? 2 ಫಾಸ್ಫೇಟ್. ಸೊ ಏನ್ ಫಾರ್ಮ್ ಆಯ್ತು? ribulose biphosphate. Now ribulose biphosphate, what is it doing? It is accepting your carbon dioxide to get converted into what? 3-phosphoglycerate. Did you understand? Now 3-phosphoglycerate is nothing but a first stable compound. It's a 3-carbon compound. Now again what is happening? Your 6 ATP is getting converted into ADP. Same. So inorganic phosphate liberate ಆಯ್ತು. ಇಲ್ಲಿ ಒಂದೇ ಫಾಸ್ಫೇಟ್ ಇದ್ದು, ಇಲ್ಲಿ bisphosphoglycerate ಆಯ್ತು. Okay na? Now it is undergoing what? Reduction. Okay, where it will form glyceraldehyde 3-phosphate nakkai glycolysis. So glycolysis ಅಲ್ಲಿ ನಿಮಗೆ ಏನಾಗುತ್ತೆ? glyceraldehyde 3-phosphate form ಆಗುತ್ತಲ್ಲ ಮಕ್ಕಳೇ. So remember that. After that it will what will happen? The regeneration of the ribulose monophosphate will happen. So this is the entire cycle of your Calvin cycle. Next one is your C4 pathway. Okay, so C4 pathway will usually take place in mesophyll and the bundle sheath cells. Am I clear to you? Now this pathway is usually method of the plants of conversion of atmospheric carbon dioxide into 4 carbon compounds. Usually the first stable compound formed here is a 4 carbon compound. Hence we call it a C4 pathway. Usually the Kranz anatomy iro plant like your sugarcane, maize, millets, papyrus, sorghum, ಇದೆಲ್ಲ usually ನಿಮಗೆ ಏನಾಗಿರುತ್ತೆ? Your C4 pathway happening. Am I clear to you? So let's start up. Now which is the first stable compound? It is your oxaloacetic acid, which is a 4 compound. ಒಂದು ನಿಮಿಷ ಮಕ್ಕಳು. Now see children, in C4 cycle, your C4 cycle is also called as your Hatch and Slack pathway. ಏನಂತ ಕರಿತೀವಿ ಮಕ್ಕಳೇ? Hatch and Slack pathway. They are the one who actually presented it. Am I clear to you? Now listen up. What will happen? Clean up case for rock. This one is nothing but your mesophyll chloroplast. This is nothing but your bundle sheath chloroplast. ಏನ್ ಮ್ಯಾಮ್ ಹಿಂಗಂದ್ರೆ ನಾವು ಕೇಳಿಸ್ಕೊಳ್ಳಿ ಇವಾಗ. I'm sure you would have seen this epidermis. Okay, I'm sorry children, it's coming very bad. Kindly adjust ಮಾಡ್ಕೊಡ್ರೋ. ಲೈಫಲ್ಲಿ ನೀವೇನೇನೋ ಅಡ್ಜಸ್ಟ್ ಮಾಡ್ಕೊಂತೀರಾ, ಇದು ಅಡ್ಜಸ್ಟ್ ಮಾಡ್ಕೊಂಡು ಒಂದು 2 minutes ಕೊಡಿ, ನಾನೇ ಬರೀತೀನಿ. ಓಕೆ, ಹಿಂಗಿರುತ್ತೆ ಕಣ್ರೋ ಎಪಿಡರ್ಮಿಸು. ಓಕೆನಾ? ಕೆಳಗಡೆ ಹಿಂಗ್ ಬರಬೇಕು, ನನ್ಗೆ ಹಿಂಗ್ ಬರ್ತಾ ಇದೆ. ಓಕೆನಾ? ಸೊ ರಿಮೆಂಬರ್ ಲೈಕ್ ದಟ್. Now in between you will have a cells like this. ಯಾವುದು? mesophyll cells. 1 minute. Okay, now see, you'll have some others. You will have some other cells like this bundle sheet. Okay, hey just listen. Okay, this is your mesophyll cell, this is your bundle sheet cell. Adjust ಮಾಡ್ಕೊಂಡ್ರಾ? ಲೈಫ್ ಅಲ್ಲಿ ಎಲ್ಲ ತುಂಬಾ ಅಡ್ಜಸ್ಟ್ಮೆಂಟ್ ನಡಿಬೇಕು, ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? So coming back to the point, this is your mesophyll cell and this is your bundle sheet cells. Am I clear to you? Now what I'm telling you, ಈ mesophyll cells ಅಲ್ಲಿ ನಿಮಗೆ ಏನಿರುತ್ತೆ? You will have your particular chloroplast present. ಅದನ್ನ ಬ್ಲಾಕ್ ಅಲ್ಲಿ ಬರೀತೀನಿ, ಅದ್ರ ಜೀವನದಲ್ಲಿ ಅದು ಗ್ರೀನ್ ಅಲ್ಲಿ ಇದೆ. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? ಈ ರೀತಿ ನಿಮಗೆ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಇರುತ್ತೆ, ಈ ರೀತಿ. Am I clear to you? Okay, so you have a chloroplast here also, you have chloroplast here also, correct? So the chloroplast in your mesophyll cell, the chloroplast in your bundle sheath cells. Okay, that is what we are showing over here. Mesophyll chloroplast and a bundle sheath chloroplast. Am I clear to you? What is it? Mesophyll chloroplast and a bundle sheath chloroplast I am speaking over here. Okay, now let's start up the cycle. What is it? Phosphoenol pyruvate is is a carbon dioxide acceptor. Am I clear to you? What is it? So then it's a carbon dioxide acceptor. So once it receives the carbon dioxide, it will get converted into a 4-carbon compound called as oxaloacetate. ಇವಾಗ ಏನಾಗುತ್ತೆ, the oxaloacetate will get converted into either the malate or either the aspartate. Okay na? So either one of them, malate ಆಗಿರಬಹುದು ಅಥವಾ aspartate ಆಗಿರಬಹುದು, will get transported into your what makla? It will get transported into bundle sheath, where your malate will get converted into pyruvate, where the carbon dioxide will be liberated, means to say decarboxylation. So the carbon dioxide so produced will be going to your Calvin cycle. Now the pyruvate whichever is formed will be transported back then to your mesophyll cell. So your pyruvate will get converted into what? Phosphoenol pyruvate. Ashte kandra, phosphoenol pyruvate carbon accept ಮಾಡಿ oxaloacetate ಆಗುತ್ತೆ. So your oxaloacetate will get converted into malate and aspartate. One of it will be transported back to bundle sheath. Malate will get converted into pyruvate. Pyruvate will be transported back to mesophyll chloroplast, where your pyruvate will get converted into your phosphoenol pyruvate. All these are the same, okay? Now, photorespiration. Now, what do you mean by photorespiration? It's a biochemical process in which plants, especially under the condition of water stress, okay, oxygen inhibits the Calvin cycle and the carbon fixation portion of the photosynthesis. Okay, so basically, the photorespiration results in what? Light dependent uptake of oxygen and release of carbon dioxide. Basically, the ಜಾಸ್ತಿ carbon dioxide ತುಂಬಾನೇ liberate ಆಗುತ್ತೆ. Am I clear to you? Its end result is what? Decreases the net amount of carbon dioxide, and both photosynthesis, photorespiration works in an opposite way. ಅಂದ್ರೆ photosynthesis ಜಾಸ್ತಿ ಆದ್ರೆ photorespiration ಕಮ್ಮಿ ಆಗುತ್ತೆ. Okay. Now the factors affecting will be usually the stomata. Okay. If they remain partially closed or completely closed, the photosynthesis is, it will not happen. Am I clear to you? So these are some of the things, and if at all the plant is under the water stress, they close the stomata to prevent the particular transpiration. The other hand, it restricts the normal exchange of the gases also. Am I clear to you? So in deserts and the dry tropical areas, photorespiration is reduced due to water stress. Factors affecting, ಮಕ್ಕಳೇ ನಾನು ಡೀಟೇಲ್ ಆಗಿ ಹೇಳಲ್ಲ. ಟೆಂಪರೇಚರ್ ಜಾಸ್ತಿ ಆದಷ್ಟು ಫೋಟೋಸಿಂಥಸಿಸ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ, but up to certain limit. After that, it will inhibit. Carbon dioxide concentration is also the same. It will increase as the particular photosynthesis also increase up to what? 0.03 to 0.04. But after that, what will happen? Your carbon dioxide, the photosynthesis will become stable. At this time, if you increase it by 0.5, again photosynthesis will increase. ಅರ್ಥ ಆಗಿಲ್ಲಲ್ಲ? ನೋಡ್ರಿ, carbon dioxide ಜಾಸ್ತಿ ಆಗ್ತಿದ್ದಾಗ carbon dioxide ಜಾಸ್ತಿ ಆದಂಗೆ your photosynthesis will also increase like this. Okay. One minute, ಮಕ್ಕಳೇ. Now it will increase like this. Up to what? Up to certain limit. After that, it will decrease like this. Okay, so when you supply again a carbon dioxide, again it will increase. Okay, so this is how it keeps on increasing. Increase agi stable agutte. Matte if you give it 0.5 ಜಾಸ್ತಿ ಕೊಟ್ಟಿದ ತಕ್ಷಣ increase ಆಗುತ್ತೆ. After which it will no more increase because your carbon dioxide will become toxic to a plant. Okay, the next one is the light. Okay, so light increases, photosynthesis increases. Oxygen inhibits photosynthesis in C3 plants, but in C4 it shows a little effect. Okay, so water if increases, the photosynthesis increases. Mineral elements too. Good elements we have to require copper, chlorine, magnesium, iron and phosphorus, air pollutants and chemical compounds. Air pollutants will reduce photosynthesis. Chemical compounds will also depend. Okay, usually they decrease the photosynthesis. Now let's start up with your particular MCQs. Okay. Now the synthesis of complex organic substances from a particular simple inorganic raw material in the presence of sunlight and chlorophyll is called as dash. Reply my drop. Yes, children. Now, synthesis of complex organic substances from simple inorganic raw material. Hey, okay, let's start up the telepathy. Okay, I forgot about the telepathy. Now, let's start up. I'm giving you a half a minute, sorry, half a second. Now, Makle, read it very clearly. In the presence of sunlight and chlorophyll is called as dash, and which process? Anabolica, catabolica. Keep it in your mind and answer for it. Am I clear to you? Is it an anabolic or catabolic? Obviously, it is a making up. So it will be what, Makle? Photosynthesis, and it is your anabolic. I can draw. Moving ahead. Which one of the following correctly depicts the biochemical reaction for photosynthesis? Neet enzyme, so sunlight and chlorophyll. Okay, which is very important. So between C and D, yadu nimge particular agi enantare. The reaction, obviously, carbon dioxide. Carbon dioxide is there, water is there. After that, you have carbohydrate, oxygen. Also, your water will also be produced. Am I clear to you? So it is C612H2O, C6H12O6. Your option is D. What is the option? It is usually your D. Now, moving on to the third question. Okay. Now, coming to the third question. Visible part of electromagnetic spectrum consists of radiation having the wavelength in the range of visible electromagnetic spectrum. Yelli namge visible light jasti irutte. Usually the visible light yava range jasti irutte. Okay. Usually it is 390 to 760 nanometer. This is the range where we usually have a visible part of electromagnetic spectrum. Moving ahead. Indigo and the red regions. Okay. Indigo and the red regions of Vibgaya respectively fall in which wavelength? Okay. Now this is the wavelength particularly corresponding to your blue and the red region. So keep that in the mind and speak about it. Okay. ನೀವು ಅವಾಗಲೇ ಓದಿದ್ರಿ. ಸೊ ಅದನ್ನ ಇಲ್ಲಿ ಕೇಳ್ತಿರೋದು. Am I clear to you? Almost blue ಮತ್ತೆ red region ಕೇಳ್ತಾ ಇದ್ದಾರೆ. ಅಂದಾಜಲ್ಲಿ ನಾವು ಕೇಳ್ಕೊಬೇಕು. Am I clear to you? So usually it is a 430 to 470 and 660 to 760 nanometer. 400 to 500 ಅಂತ ಹೇಳಿದೆ ನಗ್ತಾ ಇದೆಯಾ? So ಆ range ಅಲ್ಲಿ ನೋಡಿ. Moving on to the next question, fifth one. Okay. Now the fifth one, what it says is Moll's half-leaf experiment proves that dash is essential for photosynthesis to take place. Okay. So what does this particular experiment says is, first I will tell you what is the experiment. ಈ ರೀತಿ ಒಂದು ಟ್ಯೂಬ್ ತಗೊಂಡಾರೆ ಯಾರೋ ಮೋಲ್, ಓಕೆನಾ? ಸೊ ಇನ್ಸೈಡ್ ದಟ್ he will place a particular leaf. Like this he will place. Here what will happen? He will place a KOH. Okay, so KOH ಏನಾಗಿರುತ್ತಪ್ಪ ಅಂತ ಹೇಳಿದ್ರೆ it will absorb carbon dioxide completely. 1 minute children. One minute. Now let's start up with the thing. You will have a test tube and you will have place what, Makle? You will place a particular leaf inside. Okay. Here what you will place? You will place your KOH, potassium hydroxide. Potassium hydroxide ಏನು ಮಾಡುತ್ತೆ? It will absorb your carbon dioxide completely. What it will absorb? It will absorb your carbon dioxide completely. So basically, ಇಷ್ಟು part ನು cover ಮಾಡಿಬಿಟ್ಟಿರ್ತಾನೆ ಅವನು. Okay na? So this part will be covered. Half leaf will be exposed to the atmosphere. Half leaf will be exposed inside. So in inside ಅಲ್ಲಿ ಏನಾಗಿದೆ? Carbon dioxide ಇಲ್ಲ. There is no carbon dioxide. So obviously the photosynthesis will not take place. Why? Because there is no carbon dioxide. This was an experiment done by a moles half-leaf experiment. So this proved that it requires the carbon dioxide. In require ಬೇಕಾಗಿತ್ತು carbon dioxide ಬೇಕಿತ್ತು ಅಂತ prove ಮಾಡುತ್ತೆ. Moving ahead. Who demonstrated that the green plants purify the foul air produced by breathing animals and burning candles? Sixth one, ಯಾವಾಗಲೇ ಬೆಳಗ್ಗೆ ಅದೇ ಪರ್ಟಿಕ್ಯುಲರ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡಿದ್ದು ಹೌದು ಅಲ್ವೇನ್ರೋ ಯಾರು ಮಾಡಿದ್ದು usually who demonstrated purifying of the air? Am I clear to you? So who demonstrated that green plants usually purify the foul air produced by the first one? Who was actually produced was none other than a particular Priestley. Am I clear to you? What is it, Makla? The Priestley. Now moving ahead, the next one. Which of the following scientists concluded by his experiment that green plant parts play a role in purifying the noxious air only in the presence of sunlight? Last one ಗೂ ಇಲ್ಲಿಗೂ ಸ್ವಲ್ಪ ಡಿಫರೆನ್ಸ್ ಇದೆ. ಅವನು ಬರೀ ಪ್ಯೂರಿಫೈಯಿಂಗ್ ಏರ್ ಅಂತ ಹೇಳ್ಬೋದು, ಬಟ್ ಲೈಟ್ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಲೈಟ್ ಯಾರು ಮಾತಾಡಿದ್ರು? ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಲೈಟ್, ದ ಒನ್ ಹೂ ಫಾಲೋಡ್ ದ ಪ್ರಿಸ್ಲಿ, ಹೂ ವಾಸ್ ಇಟ್? ಇಂಜನ್ ಹೌಸ್. ಅದಕ್ಕೆ ಬೇಕಂತ ಎರಡೂ ಕ್ವಶನ್ ಕೊಟ್ಟೆ, ಕನ್ಫ್ಯೂಸ್ ಆಗಲಿ ಅಂತಾನೆ. Am I clear to you? Who demonstrated that green plants purify foul air produced by breathing plants and burning candles? It was Priestley. ಆಯ್ತಾ? But you know, in the presence of light ಅಂತ ಯೂಸ್ ಮಾಡ್ತಾನೆ. ಲೈಟ್ ಇರಲೇಬೇಕು ಅಂತ. ಯಾರು ಮಕ್ಕಳೇ? The engine house. Am I clear to you? So that's an answer. Moving ahead. Eighth one. Photosynthetic pigments such as chlorophyll A, chlorophyll B, xanthophylls and carotene can be separated by which of the following techniques? Okay, so ಈ particular pigments by using which technique we usually separate them is a particular answer question. Okay, ಮಕ್ಕಳೇ usually ನಿಮಗೆ I'm sure you would have done this particular experiment or you would have seen. ನಿಮಗೆ ಒಂದು ಬೆಲ್ ಜಾರ್ ತರ ಈ ರೀತಿ ಇಟ್ಟಿರ್ತಾರೆ, where you will have a cork present on it. Okay, and ಈ ತರ ಒಂದು hook ತರ ಬರುತ್ತೆ, where you will have a paper like this dipped. Okay na? So ಈ ತರ ಡಿಪ್ ಮಾಡಿರ್ತಾರೆ. ಎಲ್ಲಿ ಡಿಪ್ ಮಾಡಿರ್ತಾರೆ ಅಂದ್ರೆ ಇಷ್ಟಕ್ಕೂ ನಿಮಗೆ ಅಸಿಟೋನ್ ಹಾಕಿರ್ತಾರೆ. ಓಕೆ, ಮತ್ತೆ ಈ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ at this point what you will do, you will just put your one little drop of leaf pigment. Okay na? So ಅವಾಗ ಏನಾಗುತ್ತೆ, it will start segregating like this. I am sure you would have seen this in your experiments. Yes or no? Practical experimental ಮಾಡಿರ್ತೀರಾ. So this is an experiment for what ಮಕ್ಕಳೇ? Paper chromatography. ಏನು ಮಕ್ಕಳೇ? Paper chromatography. Who, after conducting the experiments on purple and the green sulfur bacteria, inferred that oxygen evolved during the photosynthesis comes from water and not from carbon dioxide? So yaar prove ಮಾಡೋದು oxygen is coming from water and not from carbon dioxide ಅಂತ ಮಕ್ಕಳೇ? It was none other than a particular Van Niel. Yes or no ಮಕ್ಕಳೇ? So the Van Niel was the person who concluded about this particular experiment. Okay. Which range of wavelength is a photosynthetically active radiation? That is your PAR. Usually we use it for the photosynthesis purpose. Okay, so the answer is usually what makla? It is none other than your 400 to 700 nanometer. Avale idal tale itkolli. Usually PAR alli the plant will use hardly 2% of the PAR. Am I clear to you? Ecosystem alli odidira adana. Which of the following serves as a source of energy for chemosynthetic bacteria? Okay, so which one of the following serves as a source of energy for chemosynthetic bacteria? It is none other than your particular it's not, of course, the sun is not the source only. Okay, yakandre idu chemosynthetic andre what? It depends upon the chemical. So now whether it is organic or inorganic is the point. It is none other than an inorganic. So from the inorganic they produce your organic substances. Now phosphoenol pyruvic acid is the primary carbon acceptor in what? So then it is usually a primary carbon acceptor in what? In the C4 plants. In C3 it is PEC. Okay, phosphoenol. So what did I say? Your phosphoenol pyruvic acid is a carbon acceptor in particularly 1 minute children, sorry, it is not C4, it is C3 plants. Okay, why? Because phosphoenol pyruvate is a 3 carbon atom. Am I clear to you? So it is a, wait a second, phosphoenol pyruvate will accept your carbon dioxide to form your oxaloacetic acid. Okay, so this is your 4 carbon compound. Correct, children? It is your C4 plants. I just got confused. Okay, it is none other than your C4 plants. Am I clear to you? I repeat again, phosphoenol pyruvate will accept the carbon dioxide to form your oxaloacetic acid to form your 4 carbon compound. Whereas in the C3, it is your ribulose biphosphate, which is your carbon dioxide acceptor to form what? To form your particular the 3 carbon compound, that is your 3 phosphoglycerate. Am I clear to you? So these are the things. During the different process of photosynthesis, splitting of water is associated with, okay, the splitting of water is usually associated with inner surface of thylakoid membrane. ಆವಾಗಲೇ ನಾನು ಹೇಳಿದ್ದೆ ಥೈಲಾಕಾಯ್ಡ್ ಮೆಂಬ್ರೇನ್ ಫೋಟೋಸಿಸ್ಟಮ್ 2 ಇಲ್ಲಿದೆ. ಸೊ ಎಲ್ಲಾಗ್ತಿದೆ ಫೋಟೋಲೈಸಿಸ್ ಹೊರಗಡೆನಾ ಒಳಗಡೆನಾ? ಇಟ್ ಇಸ್ ಇನ್ಸೈಡ್. ಓಕೆ, ಸೊ ಇನ್ನರ್ ಟು ದ ಸರ್ಫೇಸ್ ಆಫ್ ದ ಥೈಲಾಕಾಯ್ಡ್ ಮೆಂಬ್ರೇನ್. Now read the following statement and select the correct option. Statement 1, photorespiration interferes with successful functioning of the Calvin cycle. Statement 2, it usually says a particular photorespiration oxidizes ribulose 1,5-bisphosphate, which is an acceptor of carbon dioxide and Calvin cycle. Now coming back to the statement 1. Now children, photorespiration interferes with the successful functioning of the Calvin cycle. Why? Because photorespiration oxidizes ribulose 1,5-bisphosphate, which is a particular acceptor of carbon dioxide in Calvin cycle. And how do you know that statement 1 and statement 2 is interconnected? Put those questions. Why, when, what, how, so, because, henceforth. Am I clear to you? So if all those things the statement 2 answers, darling, it will always be your first option. What is it? Both statement 1 and 2 are correct and it is a correct explanation. When carbon dioxide is added to PP, that is your phosphoenol pyruvate. Am I clear to you, children? What will happen? The first stable product synthesized, it is nothing but your C4. Am I clear to you? It is nothing but your oxalate will be formed. 15th one. In cyclic photophosphorylation, the electron released by the reaction center P700 is ultimately accepted by, I repeat again, children, what is it? In cyclic photophosphorylation, the electron released by a reaction center P700 is ultimately accepted by, what is it, children? The where it will be accepted, children? First, immediately where it will be accepted? It will be accepted by the reaction center P700. So option is what? Option is nothing but your C. Am I clear to what is the option, Makla? It is usually your C. Given the particular figure, find out what is A, B, C and D. I'll give you half a minute. Do it by yourself. Okay. The 17th one, what is the option anta heli nange? What is A? Now children, idu nimge usually nanu helidevalla small pigments ಕಂಡ್ರು ಅಂತ. Okay na? These are nothing but your antenna molecules. B is usually a core molecules. Am I clear to you? Now C is your reaction center, that is your P700. Okay, and D is your primary acceptor. So usually it is your A. Am I clear to you? Now ivaga nim question kelbodu, ma'am, nimge heng gottu C is P700? Question odi alli photo system 1 anta kottore. So based on that I am doing P700. Adhe ಇಲ್ಲಿ photo system 2 anta kottidre P680 anta baritide. Okay na? How many ATP and NADPH2 are respectively produced in the process of photorespiration? ATP ಎಷ್ಟು ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರುತ್ತೆ ಮಕ್ಕಳೇ? ಓಕೆ, ATP ಎಷ್ಟು ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರುತ್ತೆ? ಯೂಶುವಲಿ NADPH ಒಂದು ಅಂದಾಜಲ್ಲಿ ಹೇಳಿ ನೋಡೋಣ. ಇದು ಫೋಟೋ ರೆಸ್ಪಿರೇಷನ್, ತಲೆಯಲ್ಲಿ ಇಟ್ಕೊಳ್ಳಿ. ಫೋಟೋ ರೆಸ್ಪಿರೇಷನ್ ಅಲ್ಲಿ ಎನರ್ಜಿ ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ರಿಲೀಸ್ ಆಗುತ್ತಾ? ಅದನ್ನ ಯೋಚನೆ ಮಾಡಿ. You will release in the photorespiration. Do you think energy will be released? Actually, it is a waste process. Okay, so it is 0 and 0. Nothing will be released. Okay, now moving ahead. Absorption spectrum of chlorophyll A and shows a maximum absorption in dash and dash regions. 19th one. The absorption spectrum of chlorophyll A maximum absorption is in blue region and the red is usually a region of the light. Am I clear to you? So option is B, blue and red. Now the last question. Dash is the process of synthesis of ATP from ADP plus inorganic phosphate. The presence of light is called as ಏನಂತ ಕರಿತೀವಿ? The addition of inorganic phosphate to the ADP. What do we call it as? We call it as photophosphorylation. ಏನಂತ ಕರಿತೀವಿ ಮಕ್ಕಳೇ? We usually call it as your photophosphorylation. Okay. Okay children, with this I'm done with the class. Take care.